Welcome students. This is our last lesson on energy transduction. In this lesson we are going to see the two stages of photosynthesis. In our previous class we have seen the introduction part of photosynthesis, and as we said, when photosynthesis occurs, light energy will be absorbed by photosensitive pigments, and this light energy is used by green plants to form chemical energy. Before the process of formation of chemical energy, let's see what will happen if light falls on pigments. When light falls on pigment, electrons will be lifted to a higher energy level, which means electrons will be excited to higher energy level, and so these electrons later on fall down to acceptor pigment, and at this time they will lose energy, and this energy is used by the cell to synthesize ATP. When we look at photosynthesis in general, it has two stages: light dependent stage and light independent stage. Therefore, from these two names you can guess that light is involved on one part of the process, and it cannot involve on the second stage. In the light dependent stage, what will happen? Here we have the process that occurs on light dependent stage of photosynthesis. These two are photosystems. By the way, this reaction occurs on the surface of thylakoid membrane. And as we said, chloroplast has photosystem, and these photosystems absorb light from different wavelengths. And here we have two photosystems, photosystem 1 and photosystem 2. Photosystem 2 and photosystem 1 are arranged in this form, and in between we have electron transport chain at which electrons will pass from one carrier to the other. Then what will happen when light falls on these two pigments? When light falls on these two pigments, electrons will be excited. These electrons will pass electron transport chain until they are accepted by the last electron acceptor. In the first pathway from photosystem 2, electrons will pass to electron transport chain, and they will be finally accepted by photosystem 1. Why? Because when light falls on these two photosystems, they lose electron, and at the same time they have shortage of electron. This electron shortage for the case of photosystem 1 will be covered by the electron that is lost from photosystem 2. This electron pass until it is accepted by photosystem 1, and the electron lost from photosystem 1 will pass to the second electron transport chain, and it can be, or it is accepted by the last electron acceptor, that is NADP, and finally it will form NADPH. Here photosystem 2 has shortage of electron because there is no electron replaced for the excited part. Therefore, what will happen? Water will split into oxygen and hydrogen. It will split into oxygen and hydrogen. From your chemistry, you know that hydrogen has one proton and one electron. The protons will be accumulate in this part of the chloroplast, and the electrons will return to photosystem 1, so that the electrons will be replaced from water. Why? Because water is splitted in this process. This splitting of water by the help of light energy is known as photolysis. By now our photosystems are stable, but the electrons pass along two electron transport chains. The electron that is lost from photosystem 2, when it passes this pathway, it loses energy. it loses energy. This energy is used to form ATP from ADP and inorganic phosphate. Due to this, ATP formation in case of photosynthesis is known as photophosphorylation. This process occurs in plant cell by formation of ATP by chemosmosis, but chemosmosis is derived by the action of light energy. For that reason, the process is known as photophosphorylation. If you remember, when we talk about ATP formation in the beginning of this unit, we have said that ATP is formed in cell by two processes. One is respiration and the other one is photosynthesis. In respiration, it occurs by substrate level phosphorylation and by oxidative phosphorylation. When we come to photosynthesis, it occurs by the process known as photophosphorylation. By photophosphorylation, ATP will be formed here, and at the same time, the electron lost from photosystem 1 will be accepted by NADP to form NADPH. This NADPH is used as hydrogen donor in the second stage of photosynthesis. Therefore, during light reaction, the cell will produce different products. The first one is oxygen will be produced as a byproduct. The second one is ATP, and the third one is reduced NADP. Oxygen will be lost as a byproduct from the leaf, but ATP and reduced NADP is used for the later stage of photosynthesis. We call this process light-dependent stage of photosynthesis. So in the light-dependent stage of photosynthesis, as we seen, it occurs in the thylakoid, on the surface of thylakoid membrane, and when light falls on the surface of thylakoid membrane, electrons will excite and they will pass different carriers until they are accepted by last electron acceptors. So if electrons pass and they are accepted by last electron acceptor, what will happen? The cell will gain ATP and NADPH for the second stage. This diagram shows you the arrangement of the different components of light reaction on the thylakoid membrane. Here you have photosystem 2. When light strikes the surface of thylakoid, photosystems release electron. This electron will pass along different carriers and it will be accepted by the last carrier. The electron from photosystem 2 accepted by photosystem 1, and the electron from photosystem 1 is accepted by NADP. In this process, the electrons will pass from one carrier to the other until they produce ATP and NADPH. The formation of ATP in this case is similar with that we have seen on the case of chemosmosis. As we said earlier, ATP produced in case of photosynthesis by a process known as photophosphorylation. But it is found that there are two types of photophosphorylation. These are cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation, as its name indicates, the electrons will return back to the first donor, or the electrons will show sort of cycling with the same molecule. And in this process, the plants only produce ATP. They produce only ATP rather than reduced NADP. When we come to non-cyclic photophosphorylation, in this case, electrons will pass along the two photosystems and there is no more cycling. As you see here on thylakoid membrane, the electrons will pass from one carrier to the other and they will pass to the last carrier, and we call this process non-cyclic photophosphorylation. In non-cyclic photophosphorylation, as we said, oxygen will be produced, ATP will be produced, and reduced NADP will be produced. But in case of cyclic photophosphorylation, the electrons will pass from photosystem 1 to higher energy level, and they will pass to electron transport chain and return back to photosystem 1. That's why we call it cyclic photophosphorylation. Here you can compare the two processes in cyclic and non-cyclic photophosphorylation. For example, in cyclic photophosphorylation, electrons will leave from photosystem 1 and return back to photosystem 1. In case of non-cyclic, electrons will leave from both photosystems, but they do not return back. The first electron donor in case of cyclic is photosystem 1. In case of water is, in case of non-cyclic is water, as we said in light reaction. The last electron donor in cyclic, photosystem 1, as I have said here. Lastly, electrons will be returned back to photosystem 1. It returned to its initial point, but in case of non-cyclic, the last electron acceptor is NADP. Products are ATP in cyclic, and non-cyclic produce ATP, NADPH, and oxygen. This is how you can compare the two photosystems. And students, as we said, in light-dependent stage, the plant produce ATP and NADPH. These two products used for the second stage or for the light-independent stage of photosynthesis. So here we are going to see the light-independent stage of photosynthesis. As we said, this process occurs in the stroma of chloroplast, and it has only three stages. The first stage is this one. It is known as carbon fixation. Why? Because atmospheric carbon dioxide will join the cycle, and it becomes part of cell process. And when atmospheric carbon dioxide joins the cycle, here we have 5-carbon compound, ribulose biphosphate. And atmospheric carbon dioxide will combine with ribulose biphosphate to produce a 3-carbon compound or a 3-carbon sugar. And in this process, it is known as carbon fixation. The second stage is reduction. The 3-carbon sugar reduced into other product, but with the same number of carbon. And the third stage is regeneration of ribulose biphosphate. This is the last stage of Calvin cycle. In this cycle, as we said, we have 3 stages: carbon dioxide fixation, carbon dioxide combined in the first reaction, and this reaction is catalyzed by a type of enzyme known as Rubisco. Rubisco is the shortest name for the enzyme. It is known as ribulose biphosphate carboxylase oxygenase, longer name. And this enzyme catalyzes the first reaction of carbon fixation, or the first reaction of light-independent reaction. But if you look at the name, it says ribulose biphosphate carboxylase oxygenase. If you look at this name, the enzyme catalyzes 2 types of reaction. It shows that the enzyme catalyzes 2 types of reaction. But from our knowledge on enzyme, we know that enzymes are specific in their action. One enzyme for one reaction. But when we come to Rubisco, it is exceptional. It can catalyze both addition of carbon dioxide or oxygen to the same substrate. That's why it is unique, and we take as unique type of enzyme. The second stage is reduction of glycerate phosphate into triphosphate. In this reaction, the cell uses ATP and reduced NADP. Both ATP and reduced NADP are result of the light-dependent reaction. The third stage is regeneration of ribulose biphosphate. In this process, what will happen? If you look at the cycle, here we have 6 molecules of 3-carbon sugar. 6 molecule of 3 carbon sugar, and it become reduced into another product, but the same amount of molecule. But what will happen? From 6 molecules, 1 molecule is used as precursor for glucose formation, and the remaining 5 molecules will rearrange themselves to form the ribulose biphosphate, to form the starting material. At this point, 1 3-carbon compound is used as a precursor, so that to form 6-carbon compound glucose, the cell need 2 turns of Kelvin cycle. The cell need 2 turns of Kelvin cycle, so that 6-carbon compound will be formed. That means in order to produce 1 glucose molecule, Kelvin cycle must be undergo 2 times in cells. This is why we consider the cycle as taking the numbers double when we calculate the product. Okay, if this is so, during regeneration of ribulose biphosphate, some of the TP or triphosphate is used to form ribulose biphosphate, 5 molecules. The remaining 1 molecule is used to form organic compounds. There are 3 types of light-independent reactions in plant: C3 or 3-carbon photosynthesis, 4-carbon, and CAM. We will see each of them in brief. C3 photosynthesis occurs in those plants that live in cold environment. Due to this, the plants are also known as C3 plants. In such type of plant, Kelvin cycle occurs in mesophyll cell, and the carbon dioxide acceptor is ribulose biphosphate. As we have seen on the Kelvin cycle, ribulose biphosphate combined with carbon dioxide to form the first reaction. And the enzyme for carbon dioxide fixation is Rubisco or ribulose biphosphate carboxylase. And the first stable product is a 3-carbon compound glycerate phosphate. That's why the plants are known as C3 plant, and the photosynthesis is also known as C3 photosynthesis. When we come to C4 photosynthesis, the plants that undergo C4 photosynthesis are more found on hot areas, in hot climate, or in tropics. And the process, or the photosynthetic process, produce a 4-carbon compound in the first reaction. For that reason, it is known as C4 photosynthesis. But when we come to the Kelvin cycle, it occurs in the bundle sheath cell of the plant. In case of C3 plant, it occurs in the mesophyll cell. But students, when we look at C4 plants, they undergo Kelvin cycle and carbon dioxide fixation at different part of their cell. One of the process, for example, Kelvin cycle occurs in the bundle sheath, whereas carbon dioxide fixation occurs in the mesophyll cell. This is one type of adaptation for such plants. And carbon dioxide acceptor is phosphoenol pyruvate. But in our previous case, the carbon dioxide acceptor is glycerate phosphate. Ribulose biphosphate. And this phosphoenol pyruvate is a 3-carbon compound. The enzyme that catalyzes the process is known as PEPCO. It's not Rubisco. PEPCO or PEP carboxylase. The first stable product is oxaloacetate, a 4-carbon compound. Due to this, the process is known as C4 photosynthesis. We call the process C4 photosynthesis. Here it shows the process of C4 photosynthesis. Atmospheric carbon dioxide absorbed by mesophyll cell. It is carbon fixation, and after it has been absorbed, C4 compound will be formed. This compound diffused to bundle sheath cell and undergo decarboxylation, and this carbon dioxide is used in Kelvin cycle inside the bundle sheath cell. This is why, because bundle sheath cells have they have chloroplast without thylakoid membrane, so that they cannot undergo the process. Secondly, Rubisco is found in the bundle sheath cell, and PEPCO is found in mesophyll cell. When we compare these two enzymes, PEPCO is more, it has more affinity for carbon dioxide than Rubisco. It can easily absorb carbon dioxide from the atmosphere, so that the process occurs in two separate cells. Okay, C4 photosynthesis, as we said, occur in plants that adapt in hot environment. And what is the main point in this process is C4 plants can store carbon dioxide in their leaves or in bundle sheath cell. This is very important point, because if atmospheric carbon dioxide increases or decreases, there is no effect on C4 plants, because they already store carbon dioxide in their leaf. In addition to this, C4 plants absorb carbon dioxide through mesophyll cells. They absorb atmospheric carbon dioxide through mesophyll cells, and this is because PEPCO has higher affinity for carbon dioxide compared with that of Rubisco. Therefore, here we have the advantage of C4 photosynthesis over C3 photosynthesis. C4 photosynthesis is advantageous under low carbon dioxide concentration, because it do not depend on atmospheric carbon dioxide concentration for carbon dioxide fixation, I mean for Kelvin cycle. The second one, high light intensity. It occurs during high light intensity and high temperature. These are the advantage of C4 photosynthesis. The third photosynthesis is CAM photosynthesis, or Crassulacean acid metabolism. Crassulacean is the name for some group of plants that undergo this type of photosynthesis, and this CAM photosynthesis is similar to some extent with C4 photosynthesis, but what we can see here is instead of a spatial difference in Kelvin cycle and carbon dioxide fixation, this type of plants undergo temporal difference, or they undergo Kelvin cycle and carbon dioxide fixation at different time of the day. The plants are called CAM plants. These plants are adapted in desert environment. You can take example cactus, balas, pineapple, and so on. CAM is effective in desert plants because it separates light dependent and light independent stage in time. The plants, as you know, all plants close their stomata when the temperature is high, when the environment is dry, in order to reduce water loss. Like that of different plants, C4 plants close their stomata during daytime. This is used to prevent evaporation and transpiration, because in desert water is very short. Water is one of problem in that environment. The plants open their stomata during the night. This is important because they are able to collect carbon dioxide from their environment. Therefore, what makes a difference with that of C3 plant? These C4 CAM plants fix carbon at night and they undergo Kelvin cycle at day. They absorb or they harvest carbon dioxide at night because stomata are open at night, and they close their stomata and undergo Kelvin cycle during the daytime. That's what we call temporal separation. As you see here, at night stomata are already open so that carbon dioxide will enter to leaves. At day, stomata are closed so they can undergo Kelvin cycle. This makes CAM plants different from C4 plants. The next point that we are going to see is photorespiration. Photorespiration is process that occurs in C3 plants. It occurs in C3 plant, and shortly photorespiration means respiration that is enhanced by light. And when photorespiration occurs, the cell use oxygen as usual and release carbon dioxide. But the main point is when photorespiration occurs, carbon dioxide, the C3 plants lose continuously their ATP, their resource, but they do not produce any product for the cell. This process occurs when atmospheric carbon dioxide is low. The concentration of carbon dioxide is low. Why? Because C3 plants, as you know, they completely depend on atmospheric carbon dioxide. Therefore, this process, as we said, reduce the efficiency of photosynthesis because only one molecule of glycerol phosphate is produced from ribulose biphosphate, but if it undergoes the normal photosynthesis from ribulose biphosphate, it produces glucose. Phosphoglycolate is produced, which must reconverted into ribulose biphosphate by using ATP. Here is the main point. It uses ATP to produce useless product. It does not produce ATP or glucose, therefore it is a wasteful and useless process. In this process, mitochondria, chloroplast, and peroxisomes are involved from cells. These three organelles are involved. In case of C4 plant, there is no photorespiration. This is because, as you know, the first reason is PEPCO, the enzyme that is used for carbon dioxide fixation, has higher affinity for carbon dioxide. Therefore, at low level of carbon dioxide, this enzyme can catalyze the reaction so that they do not get a problem in carbon dioxide concentration. And it does not take oxygen. Carbon dioxide can be stored by C4 plants. As we said earlier, in bundle sheath cell, more carbon dioxide is accumulated. This is because if you look at the nature of bundle sheath cells, they are impermeable for carbon dioxide. So carbon dioxide cannot escape from the cell. Rather, it becomes accumulated within the cell. So there is no problem of shortage of carbon dioxide for C4 plants. Students, up to now we have seen the different types of light-independent reaction in different plants. Now let's see the factors that affect photosynthesis. Since photosynthesis is a reaction, there must be some factors that can affect the process of photosynthesis. When we look at the process of photosynthesis, it is generally affected by a limiting factor in a given environment. If one factor is found to be limiting, that factor can affect photosynthesis. If you look, for example, this example, in cold, bright day, what will happen? Cold represent what? Temperature. Bright represent light intensity. Okay? But in cold, bright day, the temperature is low. Therefore, in this environment, temperature is limiting factor. Or if you look at another example, warm, cloudy day. Warm indicates high temperature. Cloud indicates low intensity of light. Therefore, in cloudy day, low intensity of light is considered as limiting factor. Therefore, when we look different environments, photosynthesis can be affected by three main factors. The first one is light intensity. Light intensity affects photosynthesis because it affects the first stage of photosynthetic reaction, light-dependent reaction. And if this process affected, the number of ATP and the number of reduced NADP produced will be also affected. Therefore, high light intensity can increase rate of photosynthesis. The second one is carbon dioxide concentration, which has direct relation with the second stage of photosynthesis, the light-independent reaction, so it can reduce glucose formation. And the third one is temperature. As any enzyme, as any chemical reaction, enzyme control the process of photosynthesis, and temperature is also important in maintaining the process of the reaction, because enzymes are affected by temperature. And the last factor is wavelengths of the light that can affect the process of photosynthesis. Okay, students, up to now we have seen photosynthesis and respiration in this unit, and in this lesson we have seen photosynthesis, and photosynthesis is a process of producing organic products by using light energy. The light energy is obtained from the environment by chlorophyll. Chlorophyll absorbs blue and red portion of light, and there are two stages of photosynthesis, light dependent and light independent stage. These two stages are interrelated, and also we have seen different types of light independent reaction. These are the C3 photosynthesis, C4 photosynthesis, and CAM photosynthesis. Lastly, we have seen three factors that affect photosynthesis. This is what you can observe in the photosynthesis part of the unit. So students, you have to read your textbook in more detail, and you have to be strong, you have to be hard worker, so as to get your final goal. This is all about our study on 11th grade biology. Thank you for listening. I wish you all the best.